I'm James. I am presenting my biomedical engineering senior design project today, which I worked on with my teammates Emily, Camilla, and Sarah. And we were advised in the Younger lab by Dr. Meg Younger and Dr. Yifan Wang. The project is titled Two-Port Olfactometer for Targeted Evolution of Mosquito Host Preference to Reduce Attraction to Humans, which, if it's as much of a mouthful it is to you as it is to me, don't worry, I'll get there. So, most of you in this room probably have been bit by a mosquito before. It's kind of annoying. You might itch for a day or two. But by the looks of it, you're all still kicking. Many people are much more unfortunate. Mosquitoes are actually the deadliest animal on Earth. And that's because of mosquito-borne diseases like malaria, Zika, dengue, and they end up killing over 700,000 people every year, which is it's an insane number. I mean, come on. The primary culprit is this species called Aedes aegypti. And what's known about them is they distinctly prefer to bite humans over other animals due to a variety of factors, and some of which are still undetermined, which our project kind of aims to look at eventually. So how do we examine mosquito, kind of scent preference and behavior? Well, it's using a device called an olfactometer. The olfactometer is this kind of large apparatus, which I'll show you in a moment, that kind of diagram like this shows that you input mosquitoes. They enter a chamber with two different stimuli at the other end. In our case, one stimulus is a human forearm, and the other is a rat. And so the mosquitoes kind of float along, they choose which scent they want to go to, and they end up trapped just before that scent. The end goal with this is that for our experiment, we want to take the mosquitoes that prefer the rat, breed them, take their kids, do it again. And if you know anything about high school biology, you'll know that if you do that enough times, you'll start to see genetic population differences. And that's what we're hoping to examine by the end of things. So the Younger lab currently has an olfactometer that they use for much smaller scale behavior assays. It's about 3 feet long in total, and about 30 or 40 mosquitoes can go in at once. The thing about our experiment that we can't use this for, to do like population level evolution, you need a really high volume of mosquitoes. 40 is not going to cut it. We want more like at least 100 in each assay. And the other kind of distinct problem with this, and this is kind of just anecdotally based on what our lab members have found using this device, is that there are areas, especially within that big chamber on the left, where mosquitoes they seem to get stuck. And they hypothesized that this was really due to like pockets within that chamber that are not getting any of the airflow from either stimulus. They're just not stimulated at all. So we aim to kind of solve that. So to characterize that poor airflow, I brought this real-life olfactometer and modeled it in a CAD software called SolidWorks. And SolidWorks has this funky little tool where you can simulate airflow through a body, and you're seeing what that looks like on the right. And so the kind of light blue, white lines you're seeing, or all those lines are representing kind of air velocity within the chamber. That white and light blue shows that like there's no air movement, and so it would make sense that there's very little odorants getting in those areas. So that's what we kind of aim to solve. So I'll show you our updated design, which we ended up implementing. This kind of changes the shape of that large activation chamber to a more conical rather than a squat rectangle. And you can see on this flow simulation on the right over there, we ended up with a lot better flow through the device. It's still very low flow just because it's a huge chamber, but it's much more well spread and less pockets of like dead air. So then our project turned to, okay, well now we got to build this thing. So we did. Here it is. It's a lot bigger than the first one you saw. This is about 6 feet long. It's pretty much double the volume. And so, yeah, you see where all the way on the right, the mosquitoes kind of enter the apparatus, move through to the left to the stimuli, and get trapped in those chambers just before the stimulus chambers. Yeah, I diagrammed that all. So yeah, they move along through. Great. So we also had to rework the stimulus chambers a little bit to properly accommodate like a forearm or a rat. So this one is the forearm. You can see a little arrow location got messed up a little bit, it looks like, but there's a little port on the left end of that thing where we can pump air in, and that kind of cross-sectional hole you see through is where your arm goes in. We're still working on, not pictured at the moment is this iris diaphragm we're putting in to seal the whole system around the arm, but that will come probably Monday or Tuesday. Anyway, here's the rat chamber. The thing about this one is it's has this nice little lid with a rubber seal around it where you can take the lid off, put your rat in, and yeah, location of things got a little messed up, but you can get the idea. So yeah, we ended up being able to manufacture this device pretty well. We've tested at this point, everything works. Mosquitoes are doing what we expect them to do, so it's great. Some future directions with this work, as I explained earlier, we're going to end up with not us, but the lab will end up with two distinct groups of mosquitoes, which they can examine genetic distinctions between, and that, looking at those genetic distinctions, they'll be able to tell, the rat preferring ones have this particular gene upregulated, or this other gene totally absent. And if you can judge which one of those makes them more attracted to other animals or less attracted to humans, you can do mosquito population genome editing whole scale in the wild, which has been done and is achievable. The other thing you can identify using those genes, there's a lot of work looking at olfactory genes and matching them to the odorants that kind of stimulate the mosquitoes. So if you can identify those specific odorants, one thing you can do is develop better repellents that kind of mute those odorants or overpower them. I want to offer a big thank you to our lab, Professor Meg Younger, Dr. Yifan Wang, and PhD student Dr. Garima Kohli. Jonathan Perry is a machinist in the biology department with whom the olfactometer cannot have produced without. He's a genius, and we are not, but he helped us a lot. And I want a big thank you to my senior design teammates, Sarah, Emily, and Camilla, and also the many, many mosquitoes we used in the last 2 weeks. Hundreds. It's just ridiculous. And thank you all for listening.
I'm interested in what you were saying about the developing better repellents.
Sure.
And is there a way that you can correlate a loss of a gene or a non-expression of a gene to its matched odorant?
Yeah, so it's the lab does a bit of that actually. So, and it's kind of a lot of trial and error work. You do a specific knockout of some olfactory gene, and suddenly the mosquitoes are loving one thing or doing something differently. They use all sorts of stimuli in their olfactometer, so they can do a lot of work to say, okay, yeah, these guys kind of sugars or something over one thing or another. So yeah, it's totally possible.
Why the length of tubing between the assembly area and the feeding area, you might say?
Yeah.
It doesn't make sense to me that it be so long, because it eliminates the immediacy of the discriminating idea.
Sure, that's a good point. I think one way to explain that is that we just needed kind of this larger volume to accommodate mosquitoes. They start to behave differently if they're like too cramped together, and they start to irk each other a little bit. And the other thing is like we kind of want them, like once they make an initial decision, we want them to stay that way. One big problem they notice is that mosquitoes will like go into one of the sides a little bit and then come out and go to the other, and that random behavior is not as desirable.
Yeah, you see like lions jumping on prey, and maybe 6 or 7 of them will be on one animal, and so the I don't know, the exposure to that stimulus for all of those animals, it's like overwhelming, and maybe something else over here, a porcupine won't get any activity. So I think that I'm confused by why force them to decide early rather than at the last moment, the moment of feeding or whatever, the moment of biting.
Yeah, so we don't actually allow them to bite. There is like a screen between the stimuli and the individual. It would be a hard sell to get someone to put their arm in there, I think. But we do notice that they get right up on that screen. They're sticking the proboscis through the holes. So they're doing their damnedest to get at that host. Ton.
I wanted to clarify something. So, in terms of next steps, if you keep going with this, or if other researchers keep going with this, preventing this or making sure that mosquitoes in the wild are.
Okay, it's better now. So, in the wild, the method of eliminating mosquitoes who tend to choose humans, is that only by breeding those who don't? Or is there another way of changing their DNA? I'm sure it's the first one, but I don't know.
Yeah, so it's done by playing with natural selection a little bit. You take your like mosquitoes you're gonna inject into the large population, and you do some gene editing. You change the thing you want to do, but you also have to change something else that makes them like have some biological advantage over the wild population, so that the change you want is kind of carried by this other kind of pro-survival change. If that makes sense.
Thank you.
Awesome. Great job, James.
Thanks.